bit of a big room to have a proper discussion, so anybody who wants to move up the front, I think that's it will help us have a bit more of a proper chat. The room is a little bit too big for our purposes. We've 3 really good talks this afternoon, and at the end of those, Catherine Summermatter, who is chair of the EU FMD Biorisk Management Group, is going to give a short overview of what the EU FMD Biorisk Management Group does and how our proposal to launch a biorisk management network can help people like us to discuss the issues and to come to some solutions on this complex area. So first of all, I'd like to welcome Claire Colenutt from Pirbright to give a talk on contaminated environment as an efficient route of transmission of foot and mouth disease virus, the very definition of biocontainment challenges. So thank you very much, Claire. I'm looking forward to this.
Okay, thank you very much for the introduction. So as Erin just said, I will be presenting on the transmission of foot and mouth disease from a contaminated environment. So I obviously don't need to tell all of you that there are multiple routes of transmission for foot and mouth disease. Direct contact with infected animals is the primary route of transmission, and this is controlled by introducing restrictions on animal movement and then disposal of infected animals. We, in our studies, have been looking at indirect transmission, so transmission from contaminated fomites, and this is less easily controlled than the effects that we can put into place to control direct contact transmission. So over the course of our studies, we were looking at the role of the environment in transmission of foot and mouth. So over the course of our study, we were using cattle transmission studies in order to sort of investigate transmission from the environment. Part of this was evaluating whether we could actually detect foot and mouth from the environment in our sampling protocols. We were looking at sampling from the environment and aerosol. I won't be focusing on the aerosol bits today, just from the solid environment. But so part of the reason for looking at this is evaluating methods of sampling from the environment that we hope to potentially use as surveillance methods as well in the future. So to start off, taking you through our experimental plan, and just to say, all of these studies were carried out at CVI in Lelystad, and these experiments all use the OUK strain of foot and mouth from the 2001 outbreak. So these experiments were obviously all carried out in high containment animal facilities where we had controlled environments, so we were controlling temperature and humidity levels as we were going. So we started off with a pair of naive cattle, which we needle inoculated in the first room. These were kept until they developed clinical signs. At the point where they developed clinical signs, we introduced another pair of animals, and this facilitated a direct contact challenge. At this point, we removed, after 24 hours of this direct contact challenge, we removed both pairs from this room, which left us with a contaminated environment. The original needle inoculated animals were then removed from the study. The direct contact animals went into a clean room where they were left to develop clinical signs, and once they developed clinical signs, they again were removed from the study. This left us with 2 contaminated environments that we could use for environmental challenges. So the immediate results from these studies are obviously whether we got transmission or not. And from the first, so we did this study twice, or this experimental protocol we carried out twice. In the first environmental challenge, which was in a room that was contaminated by needle inoculated animals, in both cases we saw transmission from this room. In our second environmental challenge, so this is in an environment that was contaminated by animals that were infected by a direct contact route. In our first study, we did not see any transmission. So the 2 animals that were put into this room, we left for 2 weeks and saw no appearance of clinical signs in these animals. In our second study, we did see transmission from this challenge. So all throughout these studies, we were collecting samples from the environment. So we were taking swabs from the walls, the floor, the feed troughs, and also collecting feces from the floors of these rooms. And we were also taking samples from the animals themselves so that we could measure both emissions from the animals and then actually quantify the amount of virus that we had in the rooms at the point of challenge. So this is an example of this data. And this data is taken from the first study that we carried out, and this is the room that was contaminated by a direct contact challenged animal. And in this case, we did not see any transmission from this room. So the top two graphs show the detection of foot and mouth in environmental samples. The graph on the left-hand side is our results from qPCR, and then on the right-hand side, we've got virus isolation results. And the day 8 along the bottom is the day that was used for challenge. So as you can see, there wasn't a huge amount of live virus present in this room. The bottom two graphs are the amount of virus that we isolated through virus isolation from oral and nasal swabs from the direct contact animals. So these are the animals that we used to contaminate the environment that we then used for the challenge. And again, we didn't pick up on a huge amount of live virus in these samples, which may explain why we didn't see transmission in this case. This is similar data, but from the second study, and so this is the same room that was contaminated by the direct contact animals. This time around, we isolated a lot more live virus from our environmental samples, but also from the oral and nasal swabs that we took from the animals. And given that we saw transmission in this case, this is presumably linked to the amount of virus that we were actually picking up at the time. So then we switched back to carrying out our experiments in Pirbright, and we took this opportunity to tweak our experimental protocol slightly. We added in an extra environmental challenge. So we started in the same way. We needle inoculated a pair of animals, and then we introduced another pair to facilitate a direct contact challenge. The difference was, in this case, we used the direct contact challenge animals, and we moved them into a first clean room, and they were left in here until they developed, or at the point of developing clinical signs, and then they were moved into a second room. So from these two animals, we had two sets of environment. One environment that had the early emissions from these animals, and the second room that had the emissions from these animals when they had full-blown clinical signs. The other change we made to the protocol at this time was when we did an environmental challenge using the first room that was contaminated by the needle-inoculated cows. We left the room empty for 24 hours between removing the needle-inoculated cattle and introducing the naive pair. So again, I've got the initial did transmission occur results from these studies. In both cases, with our first environmental challenge, so this is in a room where the contaminations were provided by the animals that were needle inoculated, but then we left it for 24 hours before introducing the naive animals for a challenge. And we saw no transmission from this room in both cases. The second environmental challenge was with the early emissions from our direct contact animals, and in both cases we observed transmission. Again, with the room that was contaminated from the direct contact animals that had the full-blown clinical signs, we saw transmission in both times again. And it's worth noting that in the second experiment, when we saw the animals going down with clinical signs, it was actually really quite quickly after the challenge. So again, we were collecting environmental samples from these studies, and I just want to draw your attention at this point to not the whole graph, but the day to which the arrows are pointing, because these are the days that we carried out the environmental challenges in each case. So this is the room from both studies that we carried out that was contaminated by needle-inoculated animals, and we were able to isolate live virus from these rooms. But it's worth noting that we did not see transmission in either of the challenges that we made from this room. So this is the same data, but from the room that was the early emissions from the direct contact animals. And again, we got kind of similar titers that we were isolating in the samples that were taken, and we did get transmission from these rooms at the time. And the same data again, or similar data, but from the room that was contaminated by the direct contact animals when they had full-blown clinical signs. And again, the arrows along the bottom point to the day on which we used the room for challenge. Again, we isolated live virus from samples that were taken in the environment, and again, we saw transmission from these rooms. So the aim of these studies was to hopefully quantify the rate of transmission that we got from the environment to naive animals. There are several issues that we found with sort of trying to estimate the probability of this. First of all, despite using pairs of animals, in the course of the study, as one animal went down after challenge, we had to remove both animals from the experiment because at that point we couldn't decipher whether the second animal in the pair would have developed clinical signs of foot and mouth from the original challenge or whether from being in contact with the first animal that went down. Also, once we sort of scaled up the amount of virus that we were getting in our samples and to the whole room that the animals were exposed to, and the fact that the animals were in there for 24 hours at a time, they potentially received really quite a large dose or exposure dose. So we have modelled, Simon has modelled this data using the positive transmission events and the amount of virus that we detected in the rooms. And we can present a weak association between the amount of virus that we see and the chance of transmission, but it potentially needs a little refining. So to conclude, we have successfully demonstrated that you can get transmission from a contaminated environment in a number of different sort of contamination scenarios, as it were. We've also been able to isolate live virus from environmental samples that we've taken, which enable us to quantify or make an estimation of the amount of virus that our naive animals are exposed to. And we're hoping to be able to sort of take these methods now and use them in an endemic setting to see if we can detect foot and mouth in those sorts of environments. And then, obviously, the aim of the project is to sort of quantify the risk associated with contaminated environments. And I'd just like to acknowledge the rest of the transmission biology group at Pirbright, and also Aldo and Fedra in Lelystad, who facilitated our first couple of animal studies, which was extremely helpful of them. And then also Jose, who previously worked with us and is now at Lelystad. And obviously to the animal technicians at both institutes that helped us greatly with these experiments. Also to acknowledge our funding from DEFRA. I'll take any questions if anyone's got any.
Thank you very much, Claire. Are there any questions for Claire? Michael, you have one here.
Thank you, that was very interesting. I was trying to follow along a little bit, but I wasn't fast enough. So of all the places in the room that you tested, were there any hotspots where you would find the most virus?
So feces was the best sample, and I'm not going to speak too much about this because Emma is doing a presentation right next that actually covers all of that, so I won't steal her thunder. But yeah.
Yes, David Payton there. I'm not sure there's a microphone. Perhaps you might just project your voice, David, in the absence of a microphone.
Just really about how useful the quantitative real-time PCR is. I mean, is there does it actually, it seemed to me that you were saying that the amount of live virus somewhat correlated with the probability of transmission. But does that also correlate well with what you're measuring by quantitative RT-PCR? Because, of course, it's much easier to measure that, but is it useful, I suppose, is the question.
I mean, I suppose when we're looking at transmission, we can obviously detect the viral RNA really quite easily, but that doesn't necessarily mean that there is live virus present. So then using that as a proxy for whether transmission is going to occur is not the most helpful thing, I suppose. And also, when we were looking at samples over a week, the amount of viral RNA we were detecting doesn't decrease, it just stays fairly steady as an amount, whereas with the live virus, it does decrease. But again, I don't want to steal Emma's thunder for her presentation.
Okay, thanks there. Well, perhaps that neatly draws us on to the next presentation. So thank you again, Claire. And Emma.